That one, the first one that I just showed you, updated, this one doesn't work, sadly, but we're making other ones. We believe that this is forming an internal hydrogen bond that would have to break, and so we're making other versions that pull them apart a little bit more so that they cannot form an internal hydrogen bond, and see if that accelerates the reaction. And then more complicated versions, this is a model of a serine esterase that Mubube in my lab is making, where you've got an alcohol, an imidazole, and a carboxylic acid, and then an oxyanion group, and all laid out with the right stereochemistry and structure of everything so that they're held in a line to mimic an active site of a serine esterase. This is the most advanced project that we have. The idea is to make new drugs, so molecules that can bind protein surfaces. So a lot of proteins bind helical peptides, and what I thought is, okay, what I'll do is I'll look for bispeptides, our molecules that can present 3 groups to mimic the side chains of an alpha helical peptide. And so I've written a lot of software. It's about 150,000 lines of C++, runs in parallel, builds these things very rapidly and scores them for how well they can present functional groups in precise orientations. And this is one of the stereoisomers that it found. Here's a superposition. This protein is HDM2 or MDM2. And it's binding the p53 peptide. p53 is the policeman of the cell. One of the ways that about 50% of all cancers suppress p53 is by expressing more of this protein. So if you can make a small molecule that binds to this protein, you'd release the green peptide, the green protein, and it could then initiate apoptosis or cell cycle arrest as an anti-cancer therapy. This has been demonstrated with small molecules. So we're trying to make this. We've made this purple molecule that can bind in here as well. And the software actually generates lots of stereoisomers that can hold 3 groups in the right orientation. So we've just started synthesizing these. The first thing we did was start with one stereochemistry, one backbone shape, and then screen different functional groups that could get binding. And we got binding right away, but it was pretty weak. But we got down to about 5 micromolar before we picked a set of functional groups and we said, okay, we're going ahead with those to do something new. And that is to vary the stereochemistry of the backbone to sort of adjust the fit of these 3 groups to see which ones can best bind into the binding site. And so we went from about 23 micromolar with this stereoisomer down to this one here, which is now 400 nanomolar, tighter than the natural peptide. And all we did was we made 20 compounds. Right? Contrast this to the small molecules. You screen libraries of like 300,000 compounds to come up with one sort of low micromolar inhibitor lead compound. We found something pretty quickly with this approach. These compounds appear to get into cells. They have a fluorescent tag on them, and they're about 1,200 Daltons. So we mix them with liver cancer cells, and then after 24 hours, wash the cells out, and they're all glowing green. These are cells with mutant p53. These are cells with wild-type p53. This is a confocal image of some of these mutant or wild-type p53 cells with our compound, and the compound is all throughout the cell. So they're getting in. I'm not sure if it's active transport or passive diffusion. Passive diffusion would be very exciting. This is very new, and we're just following this up. But we did notice that with these wild-type p53 cells, there were a lot of dead cells that have come off of the surface. And I know it's tantalizing. We're excited, and we're following up on this to see if these compounds are getting in and activating p53. And then where we're going from this is we synthesize the building blocks individually. How much time do I have left? I think I'm way ahead of time.
Oh, yeah, you go.
Oh, wow. I talk too fast.
15 minutes altogether.
Oh, okay. Well, I'll be done very quickly, and we can get on. We can synthesize the building blocks, and we can put functional groups at other positions as well, not just on these nitrogens. And so then we can assemble those pieces back together again. So we can display a lot of functionality on this backbone. All these different R groups are places where we can put functionality in. And we've published synthetic approaches to all of these guys. And so we've started screening this functional group. It's the easiest one, R4. And we screen this one, and we see that we're getting sub-micromolar binding with different stereochemistries. And we've got a lot of work to do here, a lot more compounds to make to just see how far we can go with this. And that's basically it. This is my current group. They're all working on different applications for these molecules now that we can make them and put a lot of functionality on them. We're developing applications. I've had funding from the NIH, from the NSF, and now from the Defense Threat Reduction Agency, DoD, and Temple University. And with that, I'm done. I thank you, and I'd be happy to answer any questions.